moving on now. Nitrogenous base dash is unique to DNA. Nitrogenous base dash is unique to DNA. A. Uracil, B. cytosine, C. thymine, D. guanine, E. all of the above. The answer there is C. Thymine. Thymine is a nitrogenous base that you can only find in DNA. But when it gets in RNA, it now be replaced with uracil. So uracil is found in RNA, while thymine is peculiar to DNA. The next question: acid hydrolysis of DNA causes dash. Remember we talked about hydrolysis the other time. Hydro, that is water. Lysis means breakdown. Now, acid now, that means these are solvents now. The water now, what we are using now is acid. Now, acid hydrolysis of DNA causes dash. A. Depurination, B. Translation, C. Replication, D. Methylation, E. None of the above. The answer there is depurination. When you with acid, what DNA will focus is that it will break the nitrogenous base. It will remove the base from the whole nucleotide. So we have three bases. So with that, it's altering the purine. Now you remember we have the pyrimidine. So since it's focusing on the nitrogenous base, that is where the answer came from. Depurination. Depurination. So that's the answer. The answer there is A, depurination. Now, the next question here is that DNA is relatively stable in alkaline solution because it lacks dash. A, base, B, sugar, C, 2' OH group, D, phosphate, E, all of the above. The answer there is C. That's the main reason why DNA is stable in an alkaline solution. Is because it does not have an OH group, hydroxyl group, at position 2 of their own sugar. But for RNA, it has OH at that position 2, and that is, we can just say that is the weapon fashioned against them, quote and unquote. So since DNA does not have that OH at position 2, that makes it more stable in alkaline solution. So the answer there is C. Now the next question here is the distance between two adjacent base pair is dash. The distance between two adjacent base pair is dash. Remember the structure of your DNA. We said it's a double-stranded structure. So and that double-stranded, the space between them is what we are referring to now. That distance between now the base of one strand and the base of the other. What is the distance? Now the options are A 0.34 nanometer, B 3.4 nanometer, C 34 nanometer, D 0.03 nanometer, E all of the above. The correct answer there is A, which is 0.34 nanometer. That is the distance between each nitrogenous bases. At each adjacent, that's different. That is the space distance between the adjacent base pair.